Okay, back at it again. So real quick, quick review, the dominant, recessive, homozygous, and heterozygous. So dominant is going to be referred to as a capital letter or an allele. All of them are referred to as alleles, but it's a capital letter. Recessive is going to be referred to as a little letter. So in order to have a particular trait show up and it be recessive, you have to have two little t's. And so when we have two little t's, we call that homozygous. If we have two big t's, we call that homozygous. And if we have something that is different, so a big T, little t, or any other letter in the alphabet, we call that heterozygous. So dominant, recessive, homozygous, heterozygous. And I'm going to make this, hopefully, a little bit bigger, maybe. I don't know. I don't think it's going to let me. No, it's not. Okay, we're just going to roll with it. So let me scroll down a bit so we can look at some others. And I'll keep those kind of there. All right. Maybe not. Yeah. Yeah, that works. Okay. So, a physical characteristic, something that we can look at and see, is called a phenotype. So, looking at if somebody's tall or short, that's a physical characteristic. Whether somebody's hair is blonde, brown, red, that's a physical characteristic. So that's called a phenotype. So pheno for physical characteristic. Let's try this whole writing thing again. Phenotype. Okay. Now, different from the phenotype is the genotype. So genes, genetic makeup, it's the letters that we're talking about. That is the difference between phenotype and genotype. Genotype are the genes. Genotype and phenotype is the physical characteristics. Okay, so we have dominant and recessive, we have homozygous and heterozygous, and we have phenotype and genotype. Okay, so 2, 4, 6 major vocabulary that you need to understand. So, a tool that we tend to use to be able to predict the probability of particular offspring, what they're going to be, hair colors, kinds of different things that we play around with. We use Punnett squares to do that. And so we did Punnett squares really quick with the blood types, but I feel like you guys need some more practice. And so we're going to go over some of those today. So we're going to do Punnett squares here. That's a tool that we use. It's called Punnett squares. Some people call them Punnett squares. It's not Punnett, it's Punnett. Squares. Sorry, horrible handwriting. And so it's Punnett for Punnett and then squares. So that's a tool that we use to predict the probability of offspring with a particular genotype. We can also look at phenotype with that too. We'll look at the differences. And so we can have two different ways we do Punnett squares. We can do 4 squares or we could do 16 squares. If we do one with 4 squares, we're looking at one in particular trait. So we can look at how tall or short something is. We can look at seed color. We can look at pods on peas. Are wrinkled or are they smooth? We can look at the color of different flowers. When we do that, when it's just one trait, it's not anything put together, we call it a monohybrid, and mono meaning one. So a monohybrid square goes here, or a monohybrid cross rather than a square. But regardless, it's mono. M-O-N-O. I'm trying to do a square. I'm going to have to figure out how to write better on this. Maybe I should... Oh well, mono, M-O-N-O-S-Q-U-A-R-E, mono square. It works. Now, if we're looking at two traits at the exact same time, like whether somebody's tall and short and what eye color they have at the same time. Or if we're looking at whether somebody's hair color is brown or blonde with eye color. When we're doing two traits at one time, we call that a dihybrid square or a dihybrid cross, and di means two. So we're looking at two things at one time. So this is dihybrid. Hybrid cross. Okay. So what does that look like? So when you have a Punnett square with one, I do mine like this. Miss Kennedy, who did your blood typing, did it like this. Kind of is the exact same thing, but it's kind of weird. I don't know, whatever works for you. But I do them like this, and this is how you'll see them on your test. If you are doing a dihybrid square, it's 16 squares, and it's 1, 2, 4, woohoo, like this. Far more complicated. This is far more complicated than that. So we're going to go over some examples of how we can look at Punnett squares. So let's see. I'm going to flip that. And then if I could just go to, where we go. All right. So Punnett squares. Let's look at tall and short because that's super duper easy to look at. So I'm going to say big T stands for tall. I'm going to say little t stands for short. Okay. And we're going to do two parents because we're mixing traits. So two parents we're going to do, let's say the mom is heterozygous. Heterozygous, tall. And let's say dad is homozygous, short. So first, what does that even look like? So what are the alleles that go with mom? Mom is heterozygous, tall. So hetero means different, and if hetero means different, we have to use different alleles or different characters, different letters to represent her particular genotype. And so mom, heterozygous, different, tall. If I'm different, tall, mom is. It's big T and little t. Okay. Dad is homozygous, short. Homozygous means same, short. And we've indicated that little t is for short. So we're going to make dad little t. It's recessive. So the little t is recessive to the big T, and the big T is dominant over the little t. And so this is the only way that you can get short is by two or homozygous pair being recessive. You can't be short and get and have a big T, because the big T, it doesn't matter where it is, it's always going to overpower. Another thing that you need to get used to is when you have a big T and a little t, we are used to reading capital to lowercase. We're not used to reading lowercase to capital. And so as you were doing genotypes and as you're doing Punnett squares, it makes sense to put the capital first before you put the lowercase, because that's how our brain reads anyway. There's no sense in to mess that up and get your brain all confused. So always put the capital first, and then the lowercase, and everything tends to work out in our brains the way that it should. So setting up a Punnett square with this, we have a 4 square. This is just one characteristic that we're looking at, tall and short. We're not looking at height with eye color, so we're just going to do a monohybrid square. So we're looking at one characteristic. And so we have to indicate mom's alleles and dad's alleles on this Punnett square. it doesn't matter where you put mom's alleles or where you put dad's alleles, so long as you keep them together. So mom should always go in one spot, dad should always go in another spot. You shouldn't like mix match and crisscross them to where they need to go. So you could have put mom's over on this side and dad's up top. It doesn't matter. It's not going to change your outcome or your results. Now, in order to do a Punnett square, essentially what you're doing is you're dropping and you're dragging, or you're dragging over and you're filling in the square with the alleles that correspond. Kind of like, what's that? I don't even know what game that is. Connect Four, and you're trying to connect the dots. Well, you're connecting the dots in the square. I don't know. Probably a ridiculous example. But so you have here, in this square would go a big T and a little t. Again, write your capital first and not your lowercase, because that's how our brains read. And then coming down here, you have the same thing. And then filling in this one, little t, and then this one, little t, little t. So, at this point in time, you've completed your Punnett square, but you kind of know the results of the Punnett square. What does this actually mean? And so you can be asked two different questions. What are the genotypes? Genotypes with percentages, because it's 4 squares. We do percentages with Punnett squares. We do a little bit of math with these guys. And then phenotypes. Phenotypes with percentages. Okay, so genotypes. Remember, genotypes are genes. So what are our genotypes in this particular Punnett square? Take just a second and look at it. What are the genotypes? Genes. Genes are letters. We're going to use letters. And so you have 2 of them, right? You have a big T and a little t, genotype. And you have a little t, genotypes. Little t, little t. Now, out of the possible outcomes of these 2 parents with this possible offspring, how many of them, what percentage of them can be tall? Because these little guys, the big T, little t's, are going to be tall. So what the genotypes, the big T, little t, what is the percentage? It's 50%. Ew. That looks horrible. And then what is the percentage then of a little t? So the homozygous, recessive, short, what percentage do we have of those? 50%. Okay, so those are our genotypes with percents. Remember, genotype are genes. Now, if you're asked phenotypes, that means what are the physical characteristics of the possibility of these particular offspring. So, what do they look like? What do these 2 actually look like? If you could have an offspring with this, what would that look like? It'd be tall, right? Because tall, up here it says big T equals tall, little t is short. Remember, the dominant overpowers the recessive. So even though this particular offspring carries for a short, the short's not going to be expressed. The big T dominates over the little t. And so phenotype here, remember physical, what you actually should see, we have tall, as to where when we have the little t, what does that person actually look like? Short, right? So we have tall possibilities and we have short possibilities. So out of these particular parents, what are the percentages? So if I get this particular mom and this dad together and they're going to have offspring, what percentage of them are going to be tall? 50%. And then what percentage of them are going to be short? 50%. That should always add up to 100%. A little checks and balances for yourself. Okay?